Hi there. In the last video, I talked about how DNA was packaged and segregated in bacteria primarily. And in this video, I'll talk more about how DNA is packaged and segregated in eukaryotic cells. Chromosomes in eukaryotes are packaged in distinct ways depending on whether the region of DNA is being expressed or whether it's being condensed in preparation for mitosis, replication, etc. So if you start out with a piece of DNA here whose diameter is about 2 nanometers, that's first folded around octamers of histones containing 2 of the 2HA, 2H H2Bs, 2 H3s, and 2 H4s. And then those are arranged around histone 1, which kind of lines those up into a 30 nanometer filament. And then you get these loop outs that are about 300 nanometers each in length. These are folded around a central scaffold that is made up of protein and probably RNA, as well as maybe some other components. And the DNAs are then threaded out from those. And so that's what a condensed metaphase chromosome consists of. Here's another artist's rendition of that, where you have the DNA here being folded around the histones and further packaged, and then folded around there, eventually becoming the chromosomes. And then you have many of those chromosomes. For instance, those in humans, you've got 23 sets of those. Here's an electron micrograph taken with a high voltage EM transmission electron microscope, where you can see these looped out regions of whole mount chromosomes. So this is a metaphase chromosome. There's the centromere there, and then the 2 chromatids coming out and then going down further this way. This is a different view of a metaphase chromosome where all the histones have been removed. And so you can see this central scaffold, which is made primarily of protein, but probably some RNA and maybe some other molecules as well. And then the chromatin is looped out beyond that central scaffold. So you can see all of the DNA spread out. You can see some kind of splotchy areas in here, and that's because this is a composite electron micrograph, putting several pieces together so you can see the whole image. If you focus on one particular region, then here's that scaffold made of protein, RNA, and probably some other molecules. And then here are the chromatin loops that loop out from that. And if you follow each one, you can follow it out and loop around and follow it back to the scaffold. And then it is continuous with the next one over and the next one over, looping out in these long filaments, 30 nanometer filaments. Here is a diagram of a chromosome where you have the primary constriction in the middle, which is the centromere. There are also secondary constrictions like this one, which usually contain ribosomal RNA. And as I think I've indicated before, they aren't really constrictions, or it's actually where DNA is looped out, but it doesn't stain very well, so it looks like a constriction. Each one of these is in a chromatid. And then you have these loops of DNA, and they'll stain different ways with this Giemsa stain, which is the G. So in the heterochromatin areas, they will stain darkly and stain lightly in what are called euchromatic regions. So you would have these bands of light and dark areas in the chromosome. Here are the human chromosomes stained with Giemsa in this case, I think. So you can see some darkly staining areas, which are the heterochromatin, which is usually the regions that are not being expressed at the time, so the DNA is bound up, and the lightly staining areas, which are the euchromatin, where the DNA is being expressed, so it has fewer of those histone proteins and other binding proteins attached to the DNA. So just a bit about nomenclature. So this is a metaphase chromosome with two chromatids, and there's the centromere and there's a telomere on each end. And when the usual condition before replication, the chromosome looks more like this, although they're usually more spread out and dispersed. But that's also called a chromosome. So when they duplicate, they're held together. The whole thing's called a chromosome, but there are two chromatids, and then each chromatid is the same as one of these chromosomes later when they separate. When chromosomes replicate, that is, the long chromosomes in eukaryotes, all regions don't replicate at the same time. Otherwise, it would be too slow to replicate the entire chromosome within a cell cycle. So this is chromosome 21, the long arm of chromosome 21, although if you look at any of the chromosomes in humans, you get similar results. In that there are early regions that replicate early. These are lightly colored here, and then the darkest ones are replicated last, and then there are other areas in between that are sort of in the mid-S phase. Those are replicated. So you can see replication is taking place in many different locations along the chromosome during the S phase, and that's true of all chromosomes in all eukaryotes. So here's a general cell cycle with mitosis accentuated. So in an interphase nucleus at G1, there's usually a diploid amount of DNA. In some fungi and other organisms, there's a haploid number. But by G2, after the S phase, after the chromosomes have duplicated, there's a double amount of the DNA, and you can see how the nucleus has become larger. Then in prophase, going into mitosis, the chromosomes begin to condense. So the histones and other proteins are condensing those chromosomes around those scaffold proteins. And then during prometaphase, the microtubules are starting to grow or polymerize from either end of the cell. This is showing astromitosis, where you have asters at the end, although this isn't universal for all organisms. That occurs during prometaphase, which is characterized by a lot of microtubule growth. And so eventually those microtubules grow across the center of the cell, or the middle of the cell, and they also, one set attaches to the centromere at the kinetochore proteins. By metaphase, then you have kinetochore microtubules attached to the chromosome centromere from one pole as well as from the other pole, a different set. And then these polar microtubules that are extending from one pole across the middle and passing by one another in the middle of the cell. In anaphase, you get rapid depolymerization of the kinetochore microtubules, and then there are motor proteins that are pulling those chromosomes to the poles. And you also have the polar microtubules pushing the two poles of the cells apart and stretching the cell. Finally, the chromosomes begin to decondense. If the nuclear membrane had disintegrated during prometaphase, then the nuclear membrane reforms. And then after mitosis, then you get cytokinesis, which is the pinching off, usually by actin and myosin filaments in animal cells and protist cells, which separates the two cells then. So if the original cells at G1 were diploid, then you'll get two diploid cells again. If they were haploid, then you'll get two haploid cells. And this is to show what's happening with the chromosomes, a diagram of that. So in a normal diploid nucleus in the organism, you'll have two homologous chromosomes, or a chromosome pair. and then they replicate during the S phase. So then you get a duplication of each one of those chromosomes, creating these sister chromatids in the typical shape of a metaphase chromosome. During metaphase, during prometaphase, then the microtubules reach the centromere, and at metaphase, the chromosomes of the centromeres are attached to microtubules coming from each of the two poles. And then during anaphase, the sister chromatids are pulled apart. And so once they're pulled apart, then they become those homologous chromosomes again. And so after telophase, then after the separation of the two sets of chromosomes, then you have two new cells with two nuclei containing two of the pair of those homologous chromosomes in a diploid. So to get to some of the details about some of the differences between different types of mitosis, here's astromitosis, which is normally the one that people learn. And this is characteristic of animal cells, where you have a breakdown of the nuclear membrane, and then you have microtubules forming around these centrioles or asters that are microtubule organizing centers, MTOCs. And then you have kinetochore microtubules attached to the centromeres of the chromosomes, and then polar microtubules that are crossing in the middle of the cells, which will eventually push the two cell poles apart. In plants, as well as some protists, including some dinoflagellates, they have anastromitosis. So there are no asters, there are no centrioles, but there is a microtubule organizing center, which is a more or less diffuse region near the edges of the cells. Those perform the same function in that they're full of monomers of alpha and beta tubulin, as well as some other tubulin proteins. And that's where the polymerization of the tubulins into microtubules occurs. As with animal mitosis in astromitoses, the nuclear membrane in plants cells also breaks down in a similar manner, and I'll show you later that it really doesn't break down. It's there. It just disintegrates somewhat. In some organisms, however, the nuclear membrane does not break down, and I'll talk about that in a few minutes. There also are some other variances from this normal type of mitosis, and that's with cells and some plants and animals where they have holocentric chromosomes, where there is no true centromere that you see in many organisms or most organisms. But the microtubules, the kinetochore microtubules, are attached along the entire length of the chromosomes. So then they're pulled apart more or less intact, straight over to the two poles of the cell. One other difference in plants is remember, plants have a cell wall. And so while the two cells of animals can be pinched apart by the actin filaments, that doesn't happen with plant cells. You can't pinch the cell in two because of the rigid cell walls. But as with mitosis, microtubules are heavily involved. So in plant cell division, you can see here, and these are images taken actually by a graduate student that I knew when I was a master's student. And she had to sit in for hours in a cold room taking micrographs of these, but she did a really good job anyway. And so here is the nucleus in prophase. Here's prometaphase, where the microtubules are starting to form and the cell or the nuclear membrane is breaking down. Here's metaphase, and then here's anaphase, and then telophase is here where the microtubules now have broken down. The nucleus has reformed. But what happens in the middle of the cell is a new collection of microtubules forms at that point and orients cellulose synthase enzymes to build a new cell wall across the middle of that cell. And so first of all, the cell membrane and the cell wall are built across there to separate the two cells. So that's a major difference in the cytokinesis stage in plants versus animals, in that microtubules are heavily involved here, and in an animal cell they're pinched apart by actin filaments. There are some other variations on mitosis also. So in this case, the nuclear membrane breaks down, but that's not universal. In most dinoflagellates, the nuclear membrane never breaks down, and the microtubules go through the nucleus, but they don't poke holes in it. They actually go through tunnels through the nucleus, so that the nuclear membrane is always continuous. But the microtubules do eventually, they are attached somewhat to the membrane and to the chromosomes, and eventually do pull the nucleus into two pieces, taking the homologs in opposite directions to opposite poles of the cell. And this is somewhat reminiscent of how bacteria and archaea separate their chromosomes, in that the chromosomes are attached, at least in one point, to the membrane. The cell itself, in this case, not just the nucleus, but this is the nucleus over here, the cell is then stretched and pulled by growing in the center, or in some cases at the ends, and pulling those chromosomes to the poles before splitting the cell apart. So in this case, it's the cell that's being split. In this case, it's the nucleus, but they are somewhat analogous in how they separate those chromosomes. Although in separating the chromosomes within the nucleus, you're using microtubules, and over here in bacteria and archaea, they're actually using other types of proteins that in some ways have similar functions to microtubules. In some dinoflagellates, they have true astral mitosis, whereas most dinoflagellates don't. They have anastral mitosis. And so there's a centriole, which are microtubule organizing centers, and they have a set of kinetochore microtubules that attach through the membrane to the chromosomes, through the nuclear membrane to the chromosomes, and then a set of polar microtubules that cross in the center again, just like they do in animals and plants. And so it looks very much like some of the other mitoses, although the nuclear membrane never breaks down, and these polar microtubules, again, go through tunnels through the nucleus rather than disrupting the membrane. In ascomycetes, also the nuclear membrane does not break down, and essentially the microtubules, instead of being on the outside of the nucleus, and there are some out there as well, but there are microtubules inside the nucleus, and so this looks sort of like a combination between a bacterial type and a dinoflagellate type, except you have now microtubules inside the nucleus. And so you have polar microtubules in the middle that are pushing apart the two halves of the nucleus, and then kinetochore microtubules that attach to the centromere of the chromosomes. And starting with an entire nucleus, then that pretty much stretches the nuclear membrane until you separate the two membranes. Similar to how, in a way at least, how bacteria divide. But again, this is the nucleus. In basidiomycete fungi, there's another process that occurs. The chromosomes at first are actually ripped out of the nucleus, and that nucleus does disintegrate, so that goes away. However, new nuclear membranes are formed around the separating chromosomes. So you have polar microtubules, you have the kinetochore microtubules attached to the centromere of the chromosomes, and as those start to be pulled by other microtubules out in the cytosol to the poles of that cell. New nuclear membranes form around those chromosomes. If you look at the extent of the organisms that have fission versus those that have mitosis and meiosis, generally speaking, bacteria and archaea have fission, whereas most eukaryotes have mitosis, and most also have meiosis. Although there are a few cases of organisms, including some fungi and protists, especially down here in the excavata, and some of these other protists, where they still have a fission type division rather than a true mitosis, at least in some parts of their life cycles. So here you have fission here and extending somewhat into the eukaryotes. And then over here you have a couple of types of mitotic divisions where the nuclear membrane does not break down, and that extends into some fungi as well, where the nuclear membrane doesn't break down, where in the ascomycetes it does break down in the basidiomycetes. So there's a mixture of conditions in fungi. But down here, then you have, in some dinoflagellates, they don't have the nuclear membrane breakdown, and they have anastral mitosis, whereas other ones, a few at least, have astromitosis, but the nuclear membrane doesn't break down. And then in most of the other organisms, the nuclear membrane does break down, and you have either anastromytoses, such as in the plants, or astromytoses in the case of amoebozoans and animals. So there is some evolutionary signal in all of this, in that you have fission primarily in bacteria and archaea, and then some fission in these organisms, including in ascomycetes, although the fission is primarily of the nucleus, and then you have budding that is occurring in the cell itself. And then there's an elaboration into mitoses using more microtubules to do either anastral or astromitosis without nuclear breakdown, and then further elaboration, development, evolutionarily anyway, in breakdown of the nuclear membrane, especially for organisms with larger genomes, plants and animals, essentially. So the microtubules are very important in this process of mitosis, and so I want to talk specifically about a few of the stages in mitosis, starting with anaphase, where there are actually two parts of anaphase. And both of these include microtubules, but then they include motor proteins, including dyneins and kinesins. The dyneins are attached to the chromosomes at the centromere, at the kinetochore, and that's why they're called kinetochore microtubules, and there's the dynein there. And it essentially uses ATP to walk the chromosome down the microtubule as it's being depolymerized, as it moves those chromosomes towards the poles. So that's anaphase A. In anaphase B, kinesins are being used there, and they're motor proteins also that walk along the microtubule. So one end is attached to a microtubule, and the other end is attached by these walking legs, essentially the walking parts of the kinesins. And those move in one direction along those microtubules to push the two poles apart, the two poles of the cell. So there's both a movement of the chromosomes towards the poles caused by the dyneins and depolymerization of the microtubules. And at the same time, there's a movement of the microtubules pushing the two poles apart, and addition of lengths of the microtubules. So those microtubules are actually getting longer, and they're being pushed apart so that the cell can be stretched to produce two cells. So when I was talking about the cytoskeleton, I talked about microtubules and dyneins. And the dyneins here on the kinetochore then will walk using ATP towards the minus end of the microtubule, whereas the kinesins will walk towards the plus end. And as they do in both directions, they will push those microtubules apart, pushing the poles of the cell apart, stretching the cell to eventually form, in preparation of forming two cells. So there's depolymerization also of the microtubules at the kinetochore end, and polymerization of these microtubules at these ends to lengthen them, which I always thought was quite a feat. So you're depolymerizing in one place and polymerizing in another place in an adjacent section of those cells. So here are those two molecules. Remember, they are dimers. So here's the dyneins, and there are several different dyneins. And on this end, they would be holding the chromosome. And as they hydrolyze ATP, they'll then be moving these two legs of the dimer down this microtubule towards the minus end. Kinesins, on the other hand, are also dimers, and they have these 2 feet, which then use ATP to move those on and off of the microtubule, and they move towards the plus end. And again, there are many different kinesins. So in cells where the nuclear membrane does come apart or partially disintegrate, what's actually happening there? When I talked about the cytoskeleton, I also talked about these intermediate filaments called nuclear lamins. And they're the things that really hold that nuclear membrane together and also associate it with chromosomes. So in a normal nucleus, then you have the nuclear lamins. There's the nuclear membrane, which is a membrane folded over on itself. There are the nuclear pores, and there's the DNA, which is actually attached to the lamins also through other proteins. So during the later stages of prophase and the beginning of the prometaphase, the lamins are phosphorylated. And this causes them to dissociate from one another so that they don't form the long filaments anymore. But they are still attached to parts of the nuclear membrane, so they remain attached. And this frees up the DNA and also frees up the nuclear pore complexes. And so mitosis continues. You have then early telophase, when the chromosomes have already separated at anaphase. And there's dephosphorylation of the lamins. This causes the lamins to reassociate with one another and form the long filaments, and they carry the nuclear membranes along with them. And then the nuclear pores associate with those nuclear membranes, and you start reconstructing the nuclear membrane until you have completely rebuilt the 2 nuclear membranes around the chromosomes. And again, the lamins are associated with the chromatin of the chromosome through other proteins. So they are physically attached, and by the time you get a nucleus reformed, it's in a very organized conformation, so that if you try to find sequence X or sequence Y and sequence Z in several different nuclei, you'd find them in similar orientations in those nuclei. So in this view, then, I've left these little bits of membrane in there because that's actually what happens. So although it's in a micrograph, it's not clear. You can't really see big pieces of membrane. But even though it looks like these are broken down membranes, the lamins are holding different pieces of the membrane together so that they can be rapidly rebuilt after anaphase and into telophase. So if you look at an evolutionary sequence of these things, how this is developed, first there's bacterial fission, that also occurs in archaea, but also occurs in many eukaryotic cells also. So there's not a real thick line separating these two. And then you have mitosis with intact nuclear membranes and possibly meiosis going along with that. And then mitosis with a donut-like nuclear membrane where the microtubules go through tunnels through that nucleus. And meiosis is also involved, can be involved also. Then finally you have mitosis with partial dissociation of the nuclear membrane and meiosis, and mitosis with disintegration of the nuclear membrane and meiosis. So there's sort of a sequence of events that happens here. Also, if you look at meiosis, the first division in meiosis is special in that it's different from a mitotic division, and there's recombination involved, so there's sets of slightly different proteins. But mitosis, or meiosis 2, the second division in meiosis, looks a lot like mitosis, and many of the proteins are exactly the same as they are in mitosis. So it's not as if the two are completely different. Meiosis includes a mitotic-like division as well as the initial stages, which mainly are involved recombination along the chromosomes. And in higher organisms, they have a separation between a sporophyte generation and a gametophyte generation. So all of us are sporophytes, and all the plants that you see, the flowering plants, those are all sporophytes. But they do have a gametophyte generation inside of them. And so in plants, in the flower, you have a female gametophyte where meiotic divisions give rise to several haploid cells and haploid nuclei. Those then divide, so you have haploid mitosis in those, forming a multicellular haploid part of the life cycle. And so these are, in most cases, in angiosperms, you have 8 haploid cells in the female gametophyte. In the case of lower plants, such as mosses and things, then you'll have thousands of cells in the gametophyte and only a few cells in the sporophyte. But that's I won't go into that in detail, but there are variations on whether you spend most of your time life cycle in as a gametophyte or as a sporophyte. Male gametophytes are smaller, so there are fewer cells in those. So you get a meiosis where you form 4 haploid spores. Then you get mitotic divisions of those haploid cells to form 2 cells, and then eventually 3 cells, and 2 of those end up going down the pollen tube during the pollination and fertilization stages of plant reproduction. Eventually, the pollen tube grows down the style in the flower and then goes into the ovary and enters one of the ovules of the plant. The 2 sperm nuclei are released into that gametophyte. One fuses with the egg cell down at this end, and that forms the zygote. The other one, the other pollen nucleus, fuses with these 2 central cells called polar cells in the female gametophyte and initially form a triploid cell. However, that goes on to become polyploid tissue in the endosperm. And so when you're eating corn, the maize, that juicy sweet part inside is the endosperm. And that's polyploid tissue, not just haploid, not diploid, not triploid, but it's polyploid. So the zygote eventually grows into an embryo with endosperm in the embryo there, and sometimes cotyledons, depending on the type of plant. And that's the sporophyte, in that grows up to a new plant to form gametes again, and then back into the gametophyte generation. In mammals, including humans, then The gametophyte generation is quite small. It's only one cell. So they're unicellular. You have meiosis leading to one egg cell for each meiotic division and 4 sperm cells for each meiotic division. Then you have fertilization and you're back into the sporophyte or diploid part of the life cycle again. And then we are sporophytes in that we eventually form gametes to form, and that's our gametophyte generation, and back around and around. In non-vascular plants, such as algae, this is chlamydomonas, then most of the life cycle is spent as haploids or gametophyte generation, and only a very short time is spent as the sporophyte generation. So you have these haploid vegetative cells that give rise to more haploid vegetative cells of one mating type or another, differing in their flagellar proteins, and then they fuse to form a zygote eventually, which is at a 2N level, and then immediately undergo meiosis, or almost immediately, it depends on the conditions, into the haploid generation again. So there are many organisms that spend most of their time as haploids and very little time as diploids, whereas we spend most of our time as diploids and have a small haploid generation. Many true fungi have even a different stage of their life cycle. So they can spend some time as diploids, although usually it's a short time as diploids to form haploid spores. The haploid spores then germinate and form haploid hyphae or haploid mycelium. And then they join together one mating type with another. And instead of fusing to form a single nucleus, those two nuclei stay separate and divide separately in the cells. So here's the division pattern for a typical basidiomycete. You have the two separate nuclei, one from one mating type, one from the other mating type. Then those divide mitotically, so they're being pushed apart by the microtubules. One set ends up in the middle of the cell, and so you have one of the nuclei there, the dark one there, and the dark one there. The other one gets pushed out through an extension of the cell called a clamp connection. And the extension is actually pushed out by microtubules, and the two nuclei are pushed apart by microtubules as well. That extension eventually fuses with the other cell, and a wall is built between it and the old cell, and now you have the same condition where you have two of the nuclei in one cell and two of those nuclei in the other cell. And that's called a dikaryon, and they're within the group called dikarya. Ascomycetes also are dicaryons, because they have this N+N stage where they have two different types of nuclei in the cell. So the haploid spores germinate. They can produce more haploid spores from what are called conidiophores. Those are reproductive structures in the haploid, not in the diploid. Then the two opposite hyphae can join together in a process called anastomosis, forming an ascogonium that has some of each type of nucleus in them, from one mating type and another mating type. Those can eventually form dikaryotic cells off of those multinucleate cells. And those are then N+N, or the dikaryotes. At some point in time, usually after so many cell divisions and under certain temperature regimes, then you get what's called a Crozier hook formed. And these two nuclei at the end finally fuse, and so now you have a true diploid, but it doesn't stay diploid for very long. It goes to a 4N stage, so that it replicates its chromosomes and then undergoes meiosis. And so it forms two of one type of nucleus and two of the other type. But then you have haploid mitosis after that. And so normally you have an ascus, which is the reproductive structure of these ascomycetes, which has 8 spores rather than just 4 spores. And they can actually be in different arrangements. So you can get all the red ones here and all the blue ones there, or you can get red, blue, red, blue, red, blue, and things like that. So there are different arrangements based on the species and how they set up their spindles during meiosis and mitosis. Basidiomycetes and ascomycetes can have several different forms, and one is a yeast form where they bud out of one end so that the daughter cell is a little smaller than the mother cell, or they can form hyphae. In this one, I'm just showing the yeast forms. So these are showing both the chromosomes, which are just a collection of stained regions up here. You can't really see the individual chromosomes in these organisms. The chromosomes are way too small. You can view those in an electron microscope, but not by a light microscope. And then these other lines in there are the microtubules that form. And remember, the microtubules, in the case of ascomycetes and basidiomycetes, can form within the nucleus or within the partial nucleus. But in ascomycetes, they actually form inside the nucleus. So you can see the chromosomes have been duplicated, and then the microtubules begin to form and push the chromosomes to either side of the nucleus. And there are other microtubules outside of the nucleus which coordinate the position of the nucleus. Then the microtubules begin to stretch out the nucleus. It eventually separates, and then the two nuclei separate, and finally the cell separates. In basidiomycetes, it's a little different. As I said, the duplicated chromosomes are actually ripped out of the nucleus, and then the nuclear membrane eventually just disintegrates in a corner. These two groups of chromosomes are brought to one end of the bud, and then some polar microtubules begin to form, push the two sets of chromosomes apart, and as they're pushed apart, new nuclear membrane forms around each one, and eventually they're pulled and pushed to opposite poles of that cell, and then the cell divides after that. So as far as I'm aware, both of these are fairly unique among eukaryotes in that you have microtubules, in this case, forming inside of the nucleus and then pushing the nucleus apart, stretching the nucleus until it breaks apart. But then you have other microtubules outside of that coordinating where those nuclei go. And then this one where the chromosomes are pulled out of the original nucleus, and then they start to separate, and new nuclear membranes begin to form around those. And then finally you get, again, the microtubules coordinating the positions of where those nuclei go. In the dikaryotic hyphae, or mycelia, these collections of cells that form filaments, in ascomycetes you have two different nuclei. They actually divide separately. You get duplication of the chromosomes, and then formation of the microtubules inside of each one of those nuclei. They're pushed apart and pushed and pulled actually by other microtubules to each end of those cells, and then new cell walls called septa are formed. So septa are incomplete cell walls, so there's always a connection between the two cells, although in some cases it's plugged, but in many cases there's coordination or communication between the adjacent cells. In basidiomycetes, it's even a little more complex in that you start to get the two nuclei formed. The nuclear membrane does break down, and they get pulled out away from those by microtubules. And then a little extension out of one side of the cell begins to push out. And that's also pushed out by microtubules. And then you start seeing new nuclear membranes starting to form around each one of the separating chromosome groups. This set of chromosomes then winds up in the clamp connection, called the clamp connection, which then this cell wall opens up and that one closes around it. And this nucleus is then moved into the adjacent cell. So one of the nuclear divisions looks pretty much like the ascomycete one and other, haploid cells. But this one gets pushed out into this clamp connection, and that's unique among organisms, that there's this elaborate way to move or separate those other two nuclei into the adjacent cells. So the way you can usually recognize these is these have fairly smooth cell walls on the side, and these have little bumps between the cells called clamp connections. So here you can see a typical ascomycete condition where the cell walls are smooth along their contours. And then this is a basidiomycete where you can see these bumps, these clamp connections between each pair of cells, which is where one of the nuclei traveled through that little clamp connection into the adjacent cell during cell division, nuclear division, and then cell division. So another variation on division, of course, is meiosis, and it has two different parts. One is division 1 that's primarily involved with an extensive prophase that involves recombination. So you have the chromosome pairs, not only two of the chromosomes, but all four chromatids joining together, and then crossovers occurring, and then separation of those crossovers during the prophase 1. And then there's a typical prometaphase 1, metaphase 1, anaphase 1, telophase 1. So you separate those two cells, and then you go through a second division, which is much like a mitotic division, where there's a prophase, prometaphase, metaphase, anaphase, telophase, using similar proteins or the same proteins as in a normal mitotic division. Whereas these ones up here, many of the proteins, especially these ones in prophase, are unique to meiosis. Here's a typical meiosis, or a meiotic division, where you start out with a tetraploid nucleus, that is a 4N level, and then go into what's called leptotene, which is part of prophase. The chromosomes are somewhat condensed, but not as condensed as in mitosis, so they look kind of jagged, that is, zigzaggy. And then you have zygotene, where they start pairing up, and by pachytene you have all four chromatids that are tightly pressed to one another. And then in diplotene start to pull apart some of the regions that are not involved in the crossover. And then in diakinesis, all of the sections of the chromosome have been pulled apart. So you have resolution of those crossovers. And then metaphase 1 to line up the chromosomes using the microtubules to put those in the center of the cell. Anaphase 1, separation of the homologs, not the sister chromatids, but the homologous chromosomes. So that's different from mitosis also. These are the homologous chromosomes being separated, not the sister chromatids. And then into telophase, where the nucleus reforms, and cytokinesis, and then each one of those undergoes the mitotic division, mitotic-like division, with half the amount of DNA this time. And in this case, during anaphase, you do have separation of the sister chromatids, winding up with haploid cells, that is, a haploid number of chromosomes and a haploid amount of DNA. So here's a rough comparison of the two. You have mitosis over here, where there's a tetraploid amount of DNA here after DNA replication, the same over here for meiosis. And in this case, you're separating sister chromatids, and down here in meiosis, the first division separates the homologous chromosomes that have already undergone recombination back here in the early phases of the first prophase. And then in the second division, then you have separation of the sister chromatids, winding up with 4 different haploid cells in the end. And so here, this just highlights the similarity between the second cell division and mitosis. So in the case of mitosis here, you separate from a diploid cell, you separate the sister chromatids. In this case, you're starting out with essentially haploid cells and then dividing the sister chromatids so that you wind up with haploid cells in the end. So in one case, you start out up here with cells that are at 4C but 2N, and then you separate out the sister chromatids. So in the end, you have them still at 2N but at a 2C level. Over here, you start out with cells also at 2N and 4C, but then by the end, after the second division, you come out with cells at 1C, 1N levels in each case. Of course, over here, if you have haploid cells, you start out with 2C, 1N, and wind up with 1C, 1N cells at the bottom. So this is a diagram of segregation of the chromosomes during meiosis. I already had one previously in mitosis. So you start out with homologous chromosomes, duplicate that, so now you have these at a 4C2N level. Recombination brings all 4 sister chromatids together, and then you get separation in the first division, separating out the homologs. So now these are each at 2C1N, each of the cells. And then finally separate the sister chromatids so that in the end you have 1C1N cells in each case and 1C1N chromosomes. So one of the primary differences between mitosis and meiosis is that in meiosis you have a synaptonemal complex formed, and this is a protein structure in the middle of in between the chromosomes that brings the chromosomes together. And then every once in a while there's what's called a recombination nodule, which is where you get a crossover between the 2 chromosomes. So this pretty much looks like a zipper, and it zips those chromosomes together so that they're completely homologous across, and you get then a crossover in homologous regions. Usually there are some times when you get unequal crossing over at non-homologous sites, and that occurs, of course, in ribosomal regions as well as in other regions of the chromosome. But it's a very special complex that's set up early in the prophase 1 of meiosis, and then breaks down later in the prophase section of meiosis when you get diakinesis, when the chromosomes are then starting to separate. So this next section, the last section here, talks about a few other ways where you can get alterations in the amounts of DNA and chromosomes in cells. So here's the usual way that you have mitosis and meiosis. In mitosis, you start out with cells at 2C2N. 2C is the amount of DNA, 2N is the amount of the number of chromosomes. Replicate the chromosomes. Now you're at a 4C2N stage. Separate the sister chromatids, and then you're back to a 2C2N phase of the nucleus and the cell. In meiosis, you start at 2C2N, build that up to 4C2N, but in the first division, you separate the homologous chromosomes. So now you're at a 2C1N level, reduction in the number of chromosomes, but at a 2C level, a diploid amount of DNA. And then separate out the sister chromatids, and you wind up with 4 1C1N cells, haploid cells. But just as I explained in bacteria, you can have what's called endomitosis and endoreduplication, both, and I'll explain that next. So in endomitosis, what happens is you duplicate the chromosomes, have them come apart, and then keep duplicating the chromosomes. So you have multiples of C and multiples of N, and this results in polyploidy. So those Vishafaba cells that we were extracting DNA from in lab, those cells are about anywhere from 60 to 120 C, usually about 90 C. And if you look at ZMAs, that endosperm that you're eating, that sweet part, those are at about the same level, very high polyploid levels in those, mainly to make storage proteins, starches and sugars, DNA, RNA, amino acids, etc. Another way to increase the amount of DNA in a cell, mainly to produce more copies of certain RNAs and proteins, is to undergo endoreduplication. And this is similar to endomitosis, except the chromosomes don't come apart. So you form polytene chromosomes. There's polyteny. So you have the same number of chromosomes as in a diploid, but you have multiples of the amount of the genome. So multiple genomes with the same number of chromosomes. So here's an example of that. These are salivary chromosomes from insects, and you can see that they're very, very fat chromosomes with a lot of bands, which are different puffs relating to parts of the genome that are being expressed and other parts that aren't being expressed. So you can see a puff down here. So that's making large amounts of the RNA from those genes, and it's usually in places where they need a large amount of particular proteins, such as in salivary glands, to digest part of the food. Polytene chromosomes are also found in plants. One of the primary places is in this basal cell in the embryo. So this basal cell on the suspensor, this is sort of like an umbilical cord or something like that. This end down here is attached to the plant, to the mother plant, and then this is free over here. And no one knows the exact purpose of that, but it's probably producing certain compounds that are necessary to assure that this embryo develops properly. But the polytene chromosome in this case is up to 8,192 C, so a lot of DNA, probably producing high amounts of certain proteins for that developing embryo. Some polyteny is a bit different in that you do get polyteny, but certain sequences are under-replicated. So there's still a 2n number, but then there are only partial expansions of the C amount. So you can have the C going up to 100, but some sequences are only replicated to the 25 times rather than 100 times. So you can have that condition also in certain polytene chromosomes. And finally, there's a condition where you can have extrachromosomal amplification. So this occurs in amphibians, sometimes in plants, sometimes in other animal types also. And so certain sequences are amplified either on the chromosome or extrachromosomally. So you have a 2C2N number, except there are little bits of chromosomes and little bits of the genome replicated more often than others. And this is usually when you need high numbers, high copy numbers of certain genes, and it happens in some cases in ribosomal RNA genes where you need, instead of one copy, you'll need at least hundreds of copies of that. So extrachromosomal amplification is often used for those purposes. So as I said, for bacteria, these have to make sense in terms of where these little shunts are in the cell cycle. And most of these have been confirmed in most cases about exactly where those shunts occur. So in the case of endomitosis, there is some separation of the chromosomes after G2, but there's already been replication, but mitosis is skipped. So by the time you get to G1, you're already at a G2 level in the amount of DNA. It's replicated again, skip mitosis, replicated again, skip mitosis, and increase the amount of DNA, forming a polyploid cell. For endoreduplication, then to form polytene chromosomes, during the S phase, the chromosomes never fully separate, so you go back into G1, copy them again on top of one another, copy them another time, so you get polytene chromosomes. If only part of the chromosomes are replicated, so parts of it aren't, then you get under-replication of parts of it and come out with a different type of polytene chromosome. And then amplification, again, is similar to in bacteria, where certain sequences are copied over and over and over again during the S phase. Okay, so that's the end of this video on DNA packaging and segregation, primarily in eukaryotes. The previous one was on prokaryotes primarily. And then in subsequent videos, I'll talk about other aspects of DNA replication, repair, and recombination. Okay, I will talk to you later. See ya.